Understand this part? I can't get.
But what?
This phytase gene.
I also don't get it.
Yeah, me too. No.
I have no idea what you're talking about. Phytase.
Yeah. I think
So how we complete this task?
we done it all time.
Yeah.
Oh no. Let's go find Prof. Adilini. We need to seek. Let's go. Let's go.
Hello, Prof. We really need your help, Prof. We already read through this journal. I don't know how many times. Not even 100 times, but 1,000 times, Prof. But still, we can't understand this journal. Hey, what is the phytase, Prof? Can you explain more? What is the phytase? Prof?
Relax, my student, relax. I know you are very tired today.
Yes.
Really be student, but this effort you to become a researcher. Okay, phytase is an enzyme that breaks down of fatty acid to release the phosphorus. Okay, the uses is phytase in our industry is mainly is used in monogastric animal feed, where it to reduce the what we call say the supplement of organic phosphorus to the monogastric animals such as pig and poultry. And furthermore, it is also in manufacture of human food because the ability it to improve the mineral bioavailability in our system. That's the general about phytase that you all as a student need to know.
Okay. So what you have explained just now is the usage and the meaning of the phytase, right?
Yes.
Okay.
So I have another question, Prof. What is the problem that we face during transformation of phytase gene into E. coli?
The major problem, as you know, E. coli and phytase is different. Phytase we get it from the fungal. Fungal is a eukaryotic cell, while E. coli is prokaryotic cell. So the system is very different. So one major problem is that the formation of body, fusion body, where when we express the gene phytase into the E. coli, they cannot react very well where the phytase gene become insoluble in the E. coli cell, in the cytosol. So they cannot bind very well to the E. coli. That is the major problem for this application. And the second one that we call is about the according to journal that Venice and Ushasree type 2004, it says that overexpressed gene product in E. coli cannot be suitable processed by following regulated to achieve an active food conformation. Furthermore, as I said before, in E. coli and fungal, where we get the phytase, the system is different. So also in the in the codon uses also very different. So when we try to combine it together, it maybe cannot, the system cannot mix up very well. So that is maybe become one of the factors that affect the expression level of the protein
Phytase in
in coli. That we want. So that's the major problem in this. And solution that we use in this is try to use the chaperone. Because the chaperone can be faulty or the mistake or misfolding in this gene during the gene modification in phytase and the E. coli.
So with presence of chaperone, that means phytase enzyme in the E. coli can be soluble. So there are no inclusion body form. That means no inclusion body form with the presence of the chaperone.
Yes. And before that, they have tested the many experiments that have tested the lipase also from the Candida hepatica. So they can see the efficiency of this chaperone. So the solution that we try to use in our study is that we use the chaperone plasmid during the phytase gene expression in the E. coli.
Oh, now I think we can.
We have an idea how to compare the class.
Yes. So thank you very much, Prof.
Thank
You
you,
help
Prof.
us a lot.
Yeah.
Thank you, Prof.
Okay,
Bye, Prof. See you.
Bye. This is our Aspergillus niger N2-08121, where we extract our gene of interest, that is phytase gene. After that, we amplify the gene with PCR by using our design primer, that is PHYF and PHYR. In the end of the PCR process, right now we have many copies of phytase gene. We identify our size of phytase gene by using gel electrophoresis technique. The size is approximately around 1.5 kilobase pair. After we done the gel electrophoresis, we detected the amino acid sequence from the nucleotide sequence by using expensive translate tool. Then, the molecular mass of the matched peptide was determined using peptide mass.
This matched phytase gene was obtained by PCR amplification using forward primer PET MPF and reverse primer PET MPR. Then the matched phytase gene was ligated into plasmid PET20B to generate PET PHYA and PET PHY tag. The tag used is carboxyl terminal histidine tag. Recombinant plasmid PET PHYA was transformed into E. coli DH5 alpha. Same goes with recombinant plasmid PET PHYA with histidine tag was transformed into E. coli DH5 alpha at different tube.
For subcloning in P-P4X, which is the expression vector, the phytase gene were ligated to P-P4X. Then, this recombinant plasmid were transformed into E. coli JM107. The E. coli JM107 then were growed in a medium. The selected positive clone plasmid were transformed into E. coli BL21DE3 that have plasmid PGRO7, which contain the chaperone gene.
There are 4 controls included in the experiment, which are expression strain without chaperone, Then one with induction of construct PET phi A in the absence of Arabinose. Then one with chaperone induction in the absence of IPTG, and an uninduced control in the absence of both Arabinose and IPTG. After that, several analyses were done, and as a result, we know that cytosolic solubility of fungal phytase in E. coli can be improved by co-expression of chaperone. And it can also be concluded that C-terminal histidine tag may interrupt protein folding, leading to the formation of inclusion bodies.
Summary for our research paper. The problem that we face is we found out that when we express the fungal phytase in the E. coli, there are formation of inclusion body and also insoluble aggregate. To find solution for that problem, we done the research where we isolate the phytase gene from the Aspergillus niger N2-08121, and then we done the PCR to amplify it. And then the matured phytase gene we transform into the E. coli DH5 alpha with and without histidine tag. And then we express the phytase gene into the expression vector, which is PMALB4X, and transform it into the E. coli PL21DE3 with contain vector PGR07, which is plasmid that with chaperone. And then with that method, with we done analysis and assay, we found out that presence of the chaperone in the E. coli can improve the cytosolic insolubility of the fungal phytase. And then we also found out that presence of the carboxyl terminal histidine tag in the E. coli can cause the formation of inclusion body.
That's all for us. Thank you for watching.
Bye.